bacterial plasmid DNA vector carries animal gene into host. So inserting it. So a lot of things are missing here. But still they managed to make 30 sentences. 30 odd, I think. Oh, where is that? There we go. Separation of plasmid by centrifugation. So separation of plasmid, not simply centrifugation. It won't happen, right? So it will have the alkaline method of separating plasmid from the rest. Right? That's worth to mention. And after that, centrifugation and normal protocol to extract them is the same, but to get the plasmid out very clearly from the rest, is that protocol has to be mentioned, alkaline plasmid extraction method. Right? I have made the note down, right? Just have a look at those notes. So isolation of the plasmid electrophoresis, all right. Isolation of the plasmid cut with the same restriction endonuclease, right? So plasmid has to cut with the same endonuclease. So that's how we prepare the plasmid, right? And then we put the insert, joining with the DNA fragment with plasmid using DNA ligase enzyme. That is the step where our insert join with our vector, right? So then you become a recombinant plasmid. So insert the recombinant plasmid into bacterial cell, which is called transformation. So there's a protocol for transformation as well, right? Then they say culturing the recombinant bacteria to form colonies. So that's what we did. So culturing the recombinant, because you need the blue-white colony selection to get the good one. So large number of bacteria colonies, animal genes are produced. Satisfactory transform colonies identified. How do you identify satisfactory transform colonies? Because you need to pick white colonies. That's what they're making. That's what they mean by that. Okay. If you're going to memorize this, you put in trouble because you forget it. Because now you know white colonies has to be there. Right? So you pick only the white colonies. So that is the one successful transform colony. All right. Using resistant marker, yeah, without resistant marker, so you get any bacteria can go. So then it's very complicated. How can you separate transform vector? That is why vector has to introduce ampicillin resistant gene. Then others don't go, only the transform one go. But the selective media, why selective media? Yeah, because ampicillin is a selective media. If your media is let some bacteria to go and let others not to go, that is what we call selective media. So ampicillin resistant agar in the culture media, it is a selective media. Okay. Yeah, that's the meaning of selective media. Instead, you can say ampicillin media is all right. Useful animal protein are produced by recombinant bacteria. So they haven't mentioned how do you recognize whether the protein is produced or not. You can do everything else all right, but this sentence tell you want to make sure, right? Useful protein is produced in that vector. So how do you do that? How do you confirm that it is human insulin? Right? You have to confirm it is human insulin or human growth hormone, or it could be human growth hormone, or it could be human blood protein factor. So these are the ones already cloned, right? So now we have to learn a little bit about protein. So to understand what do they mean by, right? Useful proteins are produced by recombinant bacteria has to be confirmed. Otherwise, it is not recombinant. It is not recombinant DNA technology. If you do everything, it's okay, but protein is not producing. So do you call it recombinant DNA? No. If it is recombinant DNA, two different DNA come from, let's remember the first sentence, two different molecules of DNA put together come from two different species. The second sentence, so they both together make one or many proteins, right? Or expressing or the protein synthesis is, or in other words, gene is should be expressed, right? Otherwise, if you don't express, it is not, the technology is not completed. So how do you know the gene is expressed? Expressed means the insulin protein is producing. If the protein is producing, how do you know it is insulin? It could be different protein. So that is why we have to learn about it a little bit. So we call it a Western blot. Okay. Hopefully that diagram is supposed to be there. Here we go. So Western blot. Western blot is, imagine, this kind of apparatus being used. It's a gel, but it is not agarose gel. It is a polyacrylamide gel, a different kind of gel. But it is not horizontal. This time the gel is vertical, right? The sequencing gel is also like that, vertical. Then you have a mixture of proteins, right? If you get the bacteria extraction, if you get, sometimes bacteria secrete the proteins out, right? They make a lot of insulin, and then they can't keep it inside the cell, they put it out, right? So then if you take the solution or the media the bacteria grow, so you can find that insulin in there. But insulin not there by itself. Of course, bacteria want their food want, so they secrete other enzymes as well to digest their food. So then, if you get a mixture, I mean, if you get the culture media where the bacteria are growing, so like this one, you get number of proteins in there. But now, how do you going to separate these? So you separate this by Western blot. Western blot is loading these proteins from top, and it is also, right, uh, just running through the gel, right, running through the gel. So while it's running through, it separates, making bands based on their fragments or size of the protein. Right? So, but here, right, you can identify these proteins. So one thing is the size. Right? If it is size, you have to check the size. How big the insulin? How big the insulin protein? What is the size of the insulin protein? With a marker, you can identify that. Literature will provide you information, right? So you can match with other insulin producing material and see how the fragment size. So you can Google and find out. And fragment size match is one evidence. It is insulin, right? And the second, more precise information to confirm with the insulin is kind of ELISA test, right? Kind of antibody test. So you stain the gel while you stain the protein insulin while it is inside the gel. How do you do that? Yeah, you can make anti-insulin. How do you do that? So you inject insulin into rats or rabbits, right? And draw the blood from them and remove the blood cells and take the blood plasma You don't have to purify that, but that blood plasma contain anti-insulin antibody. Right? You can inject already made insulin, or either you can made already confirm sample of insulin from high grade high grade pure sample of insulin from pharmaceutical companies to inject them into rats or rabbits. So then within 2 weeks, you can analyze the rabbit antibody level going high. So this antibody contain other normal housekeeping antibody, the rabbit has to, if any infection or any day-to-day based infection everybody get, so we have certain amount of antibody. So that is not insulin antibodies, other antibodies. But insulin antibody, once you inject insulin, then the rabbit start to generate the anti-insulin antibody, right? So you can extract antibody different protocol. So that you put the antibody in there. So your anti-insulin antibody go and attach to the exactly the band, right? Exactly the band where insulin. And then you have another antibody which is ready-made antibody. Go and bind to any antibody. So once you apply anti-insulin antibody, you wash the gel. Once you wash the gel, all the unwanted antibody was stopped. Unless very specific antibody attached to that, right? Very specific, like hybridization. Very specific antibody attached onto the gel, onto the particular band attached. So then secondary, you have a secondary antibody which go and attach to any antibody, right? And which has a marker. So you have different color markers you can have. So likewise, you give a color. So because these 3 or 4 different proteins, so they use 3 different antibodies, right? And also they use 3 different color molecules. So likewise, you can actually mark the exact protein inside the gel. You can identify that. So that is why they mention at the end in one sentence. They mention, right? One last sentence, right? Useful animal products are produced by the recombinant bacteria, right? So we have to confirm that. They don't say how, but one sentence, you can actually split this sentence and make more sentences, but 30 is enough, I think. Then, yeah, but what do you so name? It is insulin confirmed by Western blot followed by the immunity-based method, right? So yeah, you can make it better. All right, all good. So